All right, today we're going to talk about how to analyze bulk RNA-seq and ATAC-seq data and how to integrate the two assays. We will start from a FASTQ file and talking about the alignment, and then we're going to briefly talk about the RNA-seq pipeline, how to generate row count, normalize, and then perform a differential analysis to detect genes that are different between the different groups, and then pathway analysis. Then we will more focus on the ATAC-seq analysis in Partek flow and how to detect the enriched regions, and for the peaks, how to annotate peaks, visualize peaks, compare the peaks across the samples or across different groups. And then once you have a significant regions of the enriched peaks or enrichment, and we're going to do motif detection, and at the very end is pathway analysis. So last, we're going to integrate those two assays, perform correlation analysis. Right. So this data set has 32 samples. This is a subset from a geodata. I just try to make it smaller enough and also can tell the story. So they take every sample into two assays. One is they perform ATAC, one is they perform RNA. And there are two different tissue samples, tumor versus normal. So the goal in the end is try to detect the difference between tumor versus normal. So whenever you have this kind of data, you have one sample, run two assays, you should have one attribute that will pair up the two assays. For example, this column, I call that an assay pair. I have a column here. This is the ATAC-seq, and then from sample 17 to the end is RNA assays. And then this one means the assay pair means those two samples are named the same. So later on, we can pair them up. This is from the same sample. Okay, so once we have this data set up, we import the FASTQ files, and then we're going to start the analysis. For the details about how the RNA-seq data analysis, I would recommend you to go to Help, Documentation, and over here, we have some live training recordings, and I already have a bulk RNA-seq data analysis. So the detailed pipeline for RNA-seq, you can take a look at this video. So today, we're going to briefly go through the steps and then more focus on ATAC-seq analysis. Okay, so the FASTQ file import is pretty straightforward. You can either use RNA-seq or ATAC-seq to import the FASTQ file. Just select the FASTQ file. We talked about that earlier. So now, once the data is imported, just pretend you don't see the rest of the data. You only have the first data node. And when you mouse over, I have 32 samples, including both RNA and ATAC together. So the first thing I would do if you have this kind of experiment design is I want to separate the RNA and ATAC to build the pipeline in two different branches. Because I have this one column, the assay column, I can use this column, this attribute to filter my sample. So I will perform filtering, filter samples. And this is true for all the other, sample filter. If you want to exclude or include certain samples, this is not responding. Refresh that. Okay, here. So if you have attribute, it will make it easier. So I choose assay and I choose include if the assay is ATAC or RNA. So I did it twice. One is only include ATAC, one is only include RNA. That's why eventually I perform two filters to generate two data nodes. And if you right click on the column of the data node, you can rename the data node. So I can rename that, make it more meaningful. So basically, the blue color branch is my RNA data. And the green color branch is my ATAC data. You can also, right click on a task and to change the color. And when you change the color, you can change the rest. So you can do that, but as you go along, or you can do it at the very end. That's how my two different color tell me the different assays. So first, let's take a look at the RNA-seq. So within the RNA-seq, I have 16 samples, 8 tumor, 8 normal. So eventually, we're going to compare the genes to show differential expression, right? Of course, you want to perform a QA/QC. For any data, when you import it, you always want to take a look at how the data look like. And let's just take a look at it. If you double click, and this is a 100 base pair read length. And click on any of the samples. I think the data looks really good. Okay, the quality score is pretty good. And now we will perform alignment. I don't need to do any trimming. Most of the data, if you generate recently, I mean, the past few years, I think the technology is good enough. You don't have to do any training. Now for RNA-seq, because we extract the RNA, you want to use the junction aware aligners, either choose a star or HiSET. So those two aligners are for RNA-seq. The most popular one is a star. Select the aligner and choose where you want to align to. Because this is a human data, I would recommend you align to the most recent assembly, which is 38. 19 is, I think, is too old. Everybody is switched to 38 now. And you're going to pretty much just click finish. If you know what you're doing for the aligner and every time when you see the advanced configuration, you can, look at the tuning up to change it. So default should be good, and click finish. It will generate the aligner data node. And definitely, I want to check the quality. And once you click on the aligner data node, we go to post-alignment QC. I can take a look at, mainly I like to focus on the alignment rate. I think it's very good, right? There are more than 95% of the reads aligned. So the next step for RNA-seq, we will generate read count. I will perform quantification. Two popular methods. One is the Partek expectation maximization algorithm, and the other one is the public domain popular method called HT-seq. You can use either one, but the EM algorithm is much faster. So click on the task. The dialog is very much the same. You have to have a gene annotation file. So because when you align the reads to the genome, you know the read location. And now you have to tell Partek, where is your gene location? So we will know, oh, this read, if aligned to the particular location and the gene is over here, so this read is probably from this gene. And in other words, we want to summarize how many reads line up with this gene location. That way we'll generate read count. So you have to have gene annotation file. For human mouse, we distribute ensemble and gene code annotation. Whenever you have more options here, try to pick the higher number. The higher number means the more recent. If sometimes in Partek library, if they don't have a version number in their particular database, we will have a date there. Pick the most recent date when you start a project. But once you start it, don't change it, because those annotations, they keep changing all the time. And we update our annotation every 3 months. If you change it, your result will be changed. Okay, so just pick the most recent annotation. And I would say just pretty much leave everything as a default. By default, I just want to point that we only take the reads if they are 100% within the region. Okay, if the reads overhanging a transcript, we throw it away. Also, after we quantify the data, we generate read counts. We also filter out if the feature or the gene across all my samples, all my 16 samples, if the total read count is less than 10, I filter that out. So basically, some genes you might not have any reads for any of your samples. It won't. There's no point to keep all the genes. So that's why we perform a filter, and we filter it a little bit more aggressive, which means if the total is less than 10, I filter that out. But if you say, I don't want to filter anything, you can type in 0. That will give you all the genes defined in this annotation. Okay, so you can filter that later. Now, if you click finish, this annotation is based on transcript. So we generate a data node containing a transcript count. And if a gene has multiple transcripts, we will sum up all the transcript belong to this gene, and we generate a gene count. So there are 2 data nodes. If you mouse over, you will see the matrix size. 16 samples, 33,000 features, means 33,000 genes. And here, if you mouse over it, you have 145,000. Those are the transcripts. And at this point, you can click on the circle. And every time you click on a circle, you always have options to download the data. So this is a human readable matrix, and we can download. Suppose I want to put features on rows or on columns, depends on what software you want to use to open. If you use Excel, you'd better put genes on rows, because Excel cannot open this many columns. And if I download this way, it's a text file. So put it in there. And it's in a zipped text file. Once you unzip it, you can open text file in Excel. So now this matrix is, each row is a gene. We have a gene name, ensemble name, etc. From column L to column AA, this is the raw read count for each gene in each sample. And from here, you can clearly see some genes, they have a high expression, from 1,000 to 500. Some genes, they have a low expression. Some of them, they are really low. Even I did some filter already, like this one, it is very low, right? And the low expression genes usually can cause a false positive. Also, when you double click on this data node, or you can click do a task report to open that. Either way, double click, it will open the report in the current tab, browser tab. We will give you a table showing you, so this is gene level, this is transcript level. The top table is the same. Will tell you what is the percentage of the reads that is fully within your exonic region. By default, we'll only take those reads. And the bottom is showing you the summary of your matrix table, 33,000 genes, and will tell you for each gene, what's the minimum gene raw read. You're always gonna have zeros. Ideally, if the gene has no expression, they should have 0, right? But the maximum, you can see they have like 1 million reads, a mean and a median. Typically, I like to use this table to give me some idea how to filter out a low expression gene. I try to look at what is my medium look like. Medium is around 30, and the Q3 is 75th percentile, is 400 here. So I would recommend you take a look at the distribution of this table and to help you to do some unbiased filter low expression gene to increase the power for your detection. And this is like a manually binned histogram to showing you the distribution. So most of the genes, they have the reads less than 10. Each bar is a sample, and it will tell you, but we do have some between 10 to 100, etc. A box whisker plot, histogram. So based on the table, I would highly recommend you to filter out the low expression gene. But how low is low expression? Click on this data node. You can perform a filter feature, means filter genes. I would say you can pick, based on the table, I see the median is 30. Maybe you will say if the maximum value of the gene across all my 16 samples is less than 30 or, or 50. It really depends on your data. And then you can filter the low expression gene out. You're going to try it out. There's no, gold standard number how you're going to filter out the low expression gene. You can choose different parameters. If you say, oh, I want to filter out the genes if the expression value is less than such and such in what percentage of the sample. If you have a lot of samples, you can do that too. But I recommend you to use maximum as a filter. The key thing is after you click finish, always check, get the feedback. Before filter, I got 33,000 genes, and after filter, I got 17,000. Always look at the feedback to see if this is reasonable. After filter, if you only have 5,000 genes, like for human data, I think that you filtered too aggressive. You might want to redo the filter. But after you filter, you still got, 25,000 genes. I think it probably is too much, especially if you have a very low number of samples, you're going to have a hard time to pass FDR. So all data analysis is iterative, okay? So after this filter, you're going to perform normalization because the different samples they will have a different read depths. We want to make sure all the samples is on the same scale. And our default normalization is DE-seq. So you're going to choose DE-seq normalization because for most of the bulk RNA-seq, your sample size is low, and DE-seq is the most popular differential analysis method for bulk RNA-seq. Again, the normalization should go hand in hand with your differential analysis, with your downstream analysis. It depends on what you want to have what kind of distribution of the data for your downstream. If you want to do DE-seq, use a DE-seq normalization. If you want to use some other method, for example, if I want to use lemma, I would recommend you to use TMM. When you do TMM, when you build your normalization method, when you use your TMM, make sure that you're going to deal with the zero. So I would recommend you to add offset and then perform TMM. Remember, we always have zeros. Well, the DE-seq, you don't have to do it because downstream DE-seq differential analysis will deal with the zero in the differential analysis. But for the others, if you think add one is kind of too big, since we already filter out the low expression, I think add one should be fine. But if you think it's too big, you can add 0.001. And also you can perform log transformation if you will. So you can build your normalization method and click finish. In this example, and if you want to check what I did here, actually I use TMM. I will tell you the reason I use TMM because later on I want to do integration. Integration is nicer that if you, when you look at the correlation plot, it will look nicer in the log space. So that's why I didn't use the default DE-seq. That's my reason. But you can perform different normalization and then perform a different downstream analysis. I don't want to make the pipeline too complicated. It's already complicated. So I just use one, normalization for all the different downstream analysis. Okay. All right. So after we did the normalization, and you can take a look at the data and take a look at a PCA. So once you click a PCA, and then you pretty much leave everything as a default. And once you click finish, you will have the PCA data node. And if you double click on PCA, you can look at the scatter plot, look at the distribution of the data. Okay, and now I can see all the tumors, normal, they separate very well. And we know tumor and normal, they are very different. You're gonna find a lot of genes showing differential expression between tumor versus normal. If you have any other attributes like age and gender, use the style. You can also use the shape to represent, suppose represent the gender, and use the size of the dot to represent the age. So you can look at different perspective of your data. And do I see within each type of my samples, do I see the difference between male versus female or between young versus old, right? Have a bigger picture about your data. Okay. So now we're going to perform differential analysis. Since I use a different normalization method, and the lemma is a different popular differential analysis method. Our recommended is using DE-seq, but you should use the DE-seq normalization. But here, since I use TMM, I like to use lemma. They have a shrinkage. It means that they borrow variance from other genes. They are good to deal with the small number of samples. Okay, if you have a lot of samples, you can also choose ANOVA. All the dialogue is very much the same, and let's just keep it simple. I want to just compare tumor versus normal, and we just compare tumor versus normal, and I set up the comparison. Okay, and I click finish. So once this is done, the result, if I open it, the result will have, I have 17,000 genes, and this table, each row is a gene. And for every comparison, you will have the comparison tumor versus normal. We report p-value, multiple test corrected p-value, ratio and fold change is very much the same. Different presentation, and then the mean of tumor and the mean of normal, as the mean is used to calculate the ratio, and ratio converts to fold change. Even the data is in the log space, but when we give you fold change, it's always a linear space fold change. But if you say, I really want to look at a log fold change, click on the optional column, and you will have more options. And we don't call it log fold change, we call it log ratio. It's the same. So log fold change, log ratio is the fold change in log space. Okay. And you can add some other information like the chromosome location of each gene. This is all retrieved from the annotation file. From here, we can look at the volcano plot. And this is a 2D scatter plot to look at the genes. Each dot is a gene. X-axis is the fold change, but it's in the log space. Just make it look nicer. And then y-axis is negative log p-value. So basically, upper left corner and the upper right corner, those genes they have a small p-value and a big fold change. And you can select the genes what you cared about, and then take a look at what those genes are. Also, when you select that, you can label them. By default is the ensemble ID, because that's the true ID. But if you go to the style button, click on the gene name, you can change that to gene symbol, which might be meaningful to you. Okay, so this is the volcano plot. People like to present the result using this type of graph. Also, if you say, I want to focus on a particular gene, why this gene is the p-value, sorted based on p-value. You can sort it based on fold change, etc. This is the most significant genes between tumor versus normal. You click on that, you can take a look at the dot plot. On the y-axis is normalized read count in the log space, and then x-axis we separate the tumor versus normal. So we can clearly see this gene is very different between tumor versus normal. You can easily duplicate this plot and double click on the y-axis and highlight whatever the genes you cared about. If you know some gene, you can, suppose I want to search for like a EPS8, you can click on this, type in EPS8. Or if you know some genes you really like, and you can highlight that too. So if you only want to present your result just a few number of genes, you can use this function, use dot plot to present your result. Because this has the highest resolution. Once you have multiple genes displayed, you can select multiple graph by press Control and left click. Select multiple graph. You can rendering them, make the dots smaller, or you can also add box whisker on top of that and overlay that and make a color on it. So this is how you change your graph. And once you click on the export image, you can export as high resolution, and this all 4 graph together. Or if you want to focus just one, you can maximize this one and use this button to export the image for only this one gene. Okay. So this is if you want to highlight a few number for particular genes using dot plots. However, if you say, I want to select, which genes are significant, you can use the left side is a filter and specify your cutoff. And typically people will use FDR, multiple text corrected p-value. And I will say, okay, 0.01 or 0.05, since for tumor, you're going to have a lot of genes. So once you typing in the parameter, here will give you the feedback. If this is too much, you change your parameter. And also this is a p-value only tell you how significant the difference is. You can use the fold change to see how big the difference. So I want to look at the big difference. They should be at least 2-fold change or 5-fold change. You specify the parameter, and once you see this number of genes is the one I want to present, okay? And then you're going to click on the generate filtered data node. This will generate a filtered data node. I put it up right here. So I use a more aggressive filter, only have those 300 genes. And the best way to present those 300 genes to show them can differentiate the tumor versus normal, a better way to do it is using hierarchical clustering. So click on this gene list and go to exploratory analysis, and we do hierarchical clustering. This determines the order. Do you want to order your genes, features genes, right? You want to order the genes based on statistics. So the statistics, it will perform hierarchical dendrogram on it and put the genes based on the expression of the samples, and we're going to put rearrange the genes. Or if you say, I don't want to use a statistic to order the genes, I have a particular order. If that's the case, you have to make a gene list that in the order you want, and then you can choose the gene list. And the sample is the same. You can order the sample based on statistics, or you can order the sample based on, suppose I want to be based on disease, and you can change, which one is on the top or which one is on the bottom. So whichever way you want to, typically for bulk RNA-seq, you want to perform a cluster analysis. You want to see, hey, those genes really, can differentiate those tumor versus normal. So after you click finish, this is the result. See, I performed the cluster analysis. I do see this part of the genes, this section of the genes, so they are showing one behavior. This section of the genes is showing different behavior. So right now my x-axis represent the genes, and you can easily switch using the transpose to put the genes the other way, transpose them to 90 degrees. A couple things. One is if you do heat map, you can change the color of the heat map. The low expression, I can use a different color. Or you can also change the scale to see, okay, which one will help me to tell my story. So everybody has their own opinion. Whatever you think is the best to help you to tell your result. And you can also add another color. You can have multiple colors. Suppose I want to use this color represent the range over here. If there's an outlier, I want to use a different color. Okay. So this is one thing. Another thing is, the gene name. If you say, I don't want to use ensemble ID, go to axes here, determine all the labels. I can turn off the sample labels. And I want to use the gene name to label it. This might be more meaningful to present. The dendrogram, you can also color, suppose I want to color the dendrogram to highlight a different cluster. I can clearly see this group of genes is have one type of profile. This is different. And you can change the different cluster, to highlight your results. By changing this, the height of the dendrogram will give you more a space to display your heat map. Annotation is only for sample annotation. Now I'm going to overlay my disease type. Now I can clearly see all the normal samples that cluster together, all the tumor samples to cluster together. Okay, if you like to present it this way, this is how you do it. If you have too many genes, depends on your screen resolution, you might not be able to display all the genes clearly. So if that's the case, scroll your mouse wheel and to zoom in until you can see all the genes. Now each little square represents a gene. Of course, you can adjust the font of the genes, and you can right click your mouse wheel and to pan this graph. But if you want to download that, you can choose export and make sure you choose export all data. Even you don't see all the data, but once you export, it's better that you use a higher resolution. I like to use a PDF. Everybody has Adobe Reader. You can read that. You will have a higher resolution because this is really big. It's a really big picture. But no matter how you zoom in or zoom out, that's Adobe allowed me to zoom in. That's why most people, they don't present a whole lot of genes. Typically, 200 to 300 genes, and depends on your screen resolution. I'm on a laptop. My screen is not very big. So the higher resolution, the better to see the result. So this is how you present your significant gene list. Another thing is all those 300 genes, are they overly represented in a certain gene set or in a certain pathway? So if you ask that question on a filtered gene list, it have to be a subset of the gene list. And if you want to look at the biological interpretation, you will perform the gene set enrichment. You can choose a CAG database or Go database or AMSIC-DB database. Choose a database that is available to you and click Finish. The results will be like this. So this type of analysis will only take the gene name. You just count this gene happened in a unique gene name, happened in which pathway. So it's performed a chi-square. Just look at those four number of genes. Inset means in this particular genes, and I can click on this details, explain that. So this particular pathway, I have in total 24 genes. 4 genes is in my significant list. So you have to filter the list. And then 20 genes is not significant. And in my gene list, I have 119 genes, actually it's a unique ID. And 4 of them, they are in this set. 115 is not in this set. And the rest of them, they are just your background. So the total gene added up together, they're going to be the same for every pathway. So if you add up all the 4 numbers, that's the background. And within here, what you're looking for is to look at the enrichment score, which is converted from p-value. The algorithm is using Fisher exact to calculate this chi-square table. The low p-value means high enrichment score. The enrichment score, the higher the better. And FDR corrected the p-value. The rich factor means the number of the genes in the list versus not in the list in this gene set. And then that's the number of the genes. You can click on H1 to download, this which gene is in this set. Which genes in this set also is significant. Which genes is significant but not in the set. So you can download all those genes. Another visualization is you probably will see this sentence here. Say, hey, you have too many pathways. Right now, I have almost 200 pathways. It's not efficient to display that all of them. So it's better if you filter them down. Once you have less than 100 pathways in the table, you can visualize it. To filter it, one way is you can filter it right here on this table. If you say, hey, I want to look at the enrichment score, suppose greater than 3. Typically, greater than 3 is less than 0.05 of the p-value. And now you will see this data viewer. Or if you don't want to do this, another way is we have a more flexible filter. You can click on this pathway data nodes and perform a filter. So this way you can see, you can use AND or operation. You can search for a particular gene set which containing a certain keyword. Or if I want to use the same thing, I can say, hey, enrichment score greater than 3. So it's the same thing. If you click finish, that will generate a circle. But the other one does not generate a circle. But if you open the filtered list, you're going to have the same thing. You will have this view in the data viewer. Click on that. Those are all the pathways using bar chart. Everybody likes to show that. You imagine you don't want to display more than 50 of those pathways, right? So they are sorted based on the enrichment score. And another one is the dot plot. Basically each the y-axis is each pathway. X-axis you can represent rich factor, or you can click on this, say x-axis I want to represent enrichment score. So whatever you want to represent here, and then using the style, you can say, okay, I want to use the color to represent maybe rich factor, and then I can use the size to represent the genes in the set, or whatever will help you to tell your story. You can do it this way. Okay. So this is one way to interpret the biology. The other way, I would say, some of the techniques we're going to apply in the ATAC-seq also. The other one is only can be performed on the RNA-seq is GSEA. So GSEA has to be performed on the count matrix. Typically, it's normalized count matrix. The question you will ask is, I want to find a differentially expressed pathway. I don't want to look at each gene individually. I want to look at a pathway as a unit. So if you ask that question, click on the normalized count, just like when you perform a differential analysis on the gene, but you want to perform a differential analysis on the pathway. So you choose GSEA. And choose the database. Then you're going to set up, I want to compare the disease. And then look at the tumor versus normal. Set up all your comparisons and then click finish. So this result will give you each row is a pathway. And then after that, you will have a tumor versus normal. And you're going to have enrichment score. And we will recommend you to use the normalized enrichment score, because that will take the number of genes in the pathway into consideration. You can sort in either way. The sign of here will talking about which one is upregulated, which one is downregulated. So there's a sign up and down regulation information. Okay, and you can click on this enrichment graph, and this is showing you why this pathway is on the top of the list. Okay. All right, so that's pretty much what we're going to talk about for RNA-seq data analysis pipeline. And the whole point is to detect differentially expressed genes or differentially expressed pathway. All right, now let's take a look at those 16 samples in the ATAC assay. So once I got this FASTQ files, 16 samples, and you also perform pre-alignment QA/QC, and the read length is very much the same, and it looks really good too. Now we're going to perform alignment. When you have a DNA data, you are not going to use a star or a high set to perform alignment. A good option is BWA or bow tie 2. If your read length is 50, it's a very short read, you can use a bow tie. But I think nowadays everybody will use a longer read, typically 75, 100, 150, or even 200 read length. So then your option, I think the most popular one is BWA, or the other one, bow tie 2, can handle longer reads too. Bow tie 2 is faster than BWA, and you can try them both. The dialogue is very much the same. You're going to choose what assembly you're going to align to, and pretty much click finish. I will always, whether you do RNA or DNA, I will always recommend you align to the whole genome. Do not align to transcriptome, because you really don't know. When you look at the post-alignment QC, you have no idea what the alignment should be if you align to transcriptome. Okay, so use everything as a default is fine. And the post-alignment QC, I think is really good. Almost, 99% they got aligned. It's very good. And also you click on that, the post-alignment, every sample, you can see since this is a pair end, we can calculate your insert size. And typically your insert size should be longer than your read length. So I wouldn't pay attention to the peak before 100 base pair. So your sonicate fragments, it's around maybe 180 to 200 in this area. So you have some idea about the fragment length here. Now, the next thing you will do is different from RNA is I would like to filter out a duplicate, because you extract the DNA. If a read aligned to exactly the same location, and probably the read is by chance probably is a PCR artifact. For RNA, the reason you don't want to filter out duplicate reads because you don't know the RNA is the gene have a high expression or is PCR. So for R, you don't do that step, but for DNA data, you will perform filter duplicates. So let's take a look at that dialog. It's in the post-alignment QC tool. Click on the filter alignment. And the top one is to filter out duplicates. There are two options you can perform twice. One is you want to make sure I want to make the pair end the really fine pair. Sometimes when you perform the alignment, one end got aligned, the other end is not aligned. Okay, you can choose that, but I would recommend you to filter out the duplicates. So basically, if the reads, I can choose if they have the same start and same sequence. I only want to keep one. If you have more than one, that means probably it's a PCR artifact. You can keep the highest mapping score. I'm sure probably the mapping score is the same. Or unless, because they have the same sequence. Also, in this dialog, you can perform a filter low mapping quality. Mapping quality is the alignment quality. But usually the downstream analysis, for DNA data, they can take care of that. And also sometimes in a certain assays, for example, if you do cut and run and you have a certain target and you want to filter only focus on a certain location, you can choose that. And you probably will have a bad file say, hey, those are my targeted region. You can this is based on the filter sequence. And sometimes you don't want to include mitochondrial reads there because that will affect your background distribution. So you can choose to filter, exclude the reads in the mitochondrial. So this is another option. Also, this is based on certain location and say, hey, only look at the reads in a certain file. So you have to provide a bed file as annotation, and then you specify the annotation. You can choose include or exclude. But if you do whole genome, you probably don't do that. Or you can filter based on the insert size. If you say, hey, if I detected the insert size is too short or too long, I want to get rid of that. So that way you can remove some insert size. So those are the filter alignment. In this case, I only filter out a duplicate. And basically, you're going to have a less number of reads. After that, we're going to perform a peak detection. Actually, in the algorithm we have is max 3. We used to have max 2. Now they upgrade to max 3. Sorry, is peak color. Actually, within the peak color, we embedded the filter out duplicate too. It won't hurt because different algorithms, they might have different parameters to consider whether this is a duplicate or not. They might have a little bit different. So it won't hurt to perform this analysis. Or if you really want to save a step, you can just use max to filter that. And in the max 2, max 3 actually, the first thing is you specify this is a pair end or a single end. We can detect that this is a pair end. And usually they will allow you to specify the effective mapping genome size, which means in the genome, if you have some repetitive region, we won't count that as effective genome size. For human mouse, they do have this number. So you just specify that. And this part is will be a little bit different between the chip assay versus ATAC assay. For ATAC, you usually don't have like a mock or input control. You have a IP and versus your input or mock sample. So you want to use your mock sample as a background to detect. If that is the case, suppose you have 16 samples, you're going to have another 16 mock sample or input sample. So they pair them up. And using your attribute, you probably will have an attribute to pair your IP versus your input. And then that way we can generate the pair. You only detect peaks for your IP samples. But if you say only have for ATAC, you typically want to detect the peaks for all the samples. So the background is the sample itself. You don't have a separate control for that. So this part is only how you determine what samples you want to detect. So if you want to do all the samples, this is the way you generate all the samples. But if you use chip, you probably only have half of your samples. But if you don't have the attribute, you can manually say, hey, I want to manually pair that up. If I only have 2 samples, you don't have to bother to add the attribute. You just say, hey, this, suppose this is my IP sample. And this one is the corresponding, input sample. So once you click add pair, that will add, this one with this one. And we only, the control will be served as background. So if you have an enriched region in both control and IP samples, it will not call this as a peak. Okay, so that is the only difference on how to set up which sample you want to detect the peaks. And if you know what you're doing, you can configure the peak detection parameter, but I would use the default as the first iteration and click finish. After this is done, this is max result. Each row is a region. We got a lot of peaks here. Some they're, the region we have a chromosome, start and stop, and the length. You can see some length. If they are, around 200 or that mean good. If they are too long or too short, maybe they're not that great, right? They report the negative log p-value. So basically p-value is to say how confident I think that this is an enriched region compared to the background, either the neighboring in the background, or they call it local background, local lambda, or compare with your input samples. And the fold enrichment is just the peak compared to the background. Again, the higher the better. This negative log p-value is higher the better. Some people think you should consider only if the negative log p-value is greater than 10, or some even say it should be greater than 20. The optional column will give you some other information. You can get the Q value also, and the absolute summit. And the summit is the base pair location that you have the most reads here in this position. That's the sample name. So here I would like, take a look at all the peaks. Especially look at the p-value. So we can take a look at if the p-value is like the first one is 37. It's a pretty high log p-value, negative log p-value. And over here is browse to location. We call that a chromosome view. When you do DNA data, you probably want to look at the chromosome view a lot. When you do RNA, because the genes, they next to each other, they might not be correlated, right? So it's different. For DNA, you want to look at the genomic location. So let's talk about a little bit about Partek chromosome view. It's similar to IGV, but IGV you download to your local computer, and your data is on local computer, so the retrieval file will be faster. But here, all your data or all the BAM files is on the server, and the browser is on the local, so it will take a little longer time to sometimes to display the visualization. And also like a UCSC genome browser, they are very much the same function. So basically your x-axis, you look at the genomic location. And currently, Partek only allows you to look at each chromosome as each chromosome at a time. You cannot look at the whole genome. That's why we call that a chromosome view. Yeah, well, this is kind of too slow. Yeah, so let me just talk about a little more. From here, you can search a gene name, or you can search a location. The location name is the chromosome : start dash stop. So that's how you specify the location. Just did something wrong here. Okay. And on the right side is zoom in and then zoom out. So here we look at this location, and over here we group the data based on the disease. We can see one track is the normal, and the other track is the tumor. And here, this visualization might be misleading. You are thinking, oh, the blue layer S1 has more read. No, that's not. They are just stacked on top of each other. But if you see to compare each sample, what you can do is, For each track, there's a gear button. The 3-line button allows you can pin to the top or to the bottom, or you can hide that, not show. But the gear button, you can choose the configure. So the color represents the sample. You can choose, okay, I want to use the histogram. Sorry, the color you can use represent the different things, represent disease or represent different attribute, or even the base pair. But only when you zoom in to a very to a very focused location, you can see each base pair. And then the style, this is a stack on top of each other. If you choose overlay, then that'll be more is more accurate. So if I click apply, you can apply for all the tracks. That means you apply to this track also. Don't forget to apply to click this apply button. That will refresh this. You will see the different style. But the bottom here is the peaks location for each sample. And you can choose to display all the samples in one track. Maybe you want to color them based on tumor versus normal, or you can choose to look at each sample separately. So that's how you configure that. And then in the select tracks, this button allows you to select. So once you align the data in the BAM file, you have the location of the reads, you are able to draw the chromosome view. So you can display the aligned data or even RNA aligned data. And then this is a filtered alignment, and this is the peak, annotated peak, etc. So you can click on this and check whether you want to look at it or not. Also at the bottom, you can choose the gene annotation. So suppose I want to use my ensemble database, I can display the ensemble annotation. So this will take some time. In the meantime, yeah, let me just go on to do the analysis. So you might want to filter out some peaks if you say, okay, if the peak is too long, or too short, I don't consider that is the transcription factor binding site, for example, right? Or you can filter based on p-value or enrichment score. To do that, you click on this peak data node and go to peak analysis and choose to filter peaks. You can use the and or operation. So I just want to show you an example. So suppose I want to be stringent on the p-value. I will say, okay, look at the negative log p-value should be greater than or equal to 10 or 20 or I'll just maybe 15. And I want the read length. It's not too long, should be less than, suppose, 500 base pair. I don't want to look at more than 500 base pair. And if it's too short, it's not good. The read length should be greater than, since my read length is 100, so it's better, longer than your read length, maybe 150 base pair. So if you think your binding site is kind of between in this range, that's how you perform the filter. Okay, and then click finish. Let's see if okay, so this is the chromosome view. As you can see here, because I overlay, you only focus on the line. It seems like the sample 16 has the highest, has more reads because it's higher over here, right? That's in the normal samples, but in the tumor samples, this is a 15, has more reads over here. And you can also see the lower read. And the bottom is the, for each sample, we detect the region. So it seems like in this region, every sample is enriched. And also when you look at this, when you look at this, you can zoom out a little bit, because look at the neighboring is very important too. So I will just let it run. And after I filter the peaks, I got this 100, almost 200,000 regions. Because this table is huge. When you have, I forgot the threshold, a certain number of rows, less than a certain number of rows, you can sort it. But if it's too many rows, we cannot sort it. You can download it and download this as text file to output the data. Okay, so now we have all the peaks. We detect the peaks for all the samples. And then the first thing you want to know are those peaks fall into how they related to the genes, right? So the next question you will ask, I want to see the gene location. So on the peaks, you will perform annotate peaks, and you will provide a gene annotation file. So you have to tell Partek where gene location is. Because the promoter is not defined, we will ask you define it. Your TSS is, I mean, if your TSS upstream 1,000 and downstream 1,000, I will think this is the promoter region. Or you can also define the TTS upstream and downstream. So that is for you to find a peak overlap of gene. Also, you think about the peak and the gene, they are not always very clean. And in the same location, when you talking about the transcription factor, you think about the transcript level, not the gene level, right? And for one location, you might have a multiple transcript here. Let's see if my okay, so now if I zoom out, you can see, oh, this is really a peak if I look at the neighboring, right? So all the samples detect this peak is pretty good. And this is the ensemble annotation. In this location, each little gray thing is a transcript. When you see the thicker, the higher one, that means it's exon. And this thin line is intronic region. Okay, and then the arrow means is on the forward or reverse strand. So some genes, they might have 17 different transcripts. They all have the same start, stop, maybe one transcript is skip an exon or something like that, right? So when you see the region overlap with a transcript, you might have multiple, maybe overlap with this transcript, the exonic region, the other one is intronic region, right? So if that is the case, when you report, we have a two format. One is you report one gene region per peak. You only pick one thing. And if that's the case, we have to apply the precedency. So if this region overlap with the multiple genes or transcript, we first report the TSS. If this gene is on my TSS region, the other gene is on the intronic region, I will report the TSS region gene. And then TTS and the CDS exon and UTR 3', 5' intronic, and then the last is the intergenic region. Or you can choose, I report all of them. Depends what you want to do downstream. If you have one region overlap with multiple genes, if you want to download the table using some other software to do other things, it's going to be very messy. So that's why we give you two formats. Okay. So I will just leave this one here and click finish. Once you get this results, and this is the annotated peak, and we give you a bigger picture, just telling you for all the samples for all the regions, we put them all together. If you want to look at each sample separately, you might want to filter your peaks on certain sample and then do the annotation. Or after you've done annotation, you can filter for a sample that the graph will report for each sample separately. This is the bigger picture, and then the result is from this chromosome to sample name, this is what we inherited from the peak. And after that is we add all the information for the gene. From the optional column, you can add more. You can see when you do the overlap, and they will have some percentage there. So when you're talking about the region, it's really not like a gene. Every sample of the gene stop is the same. This is not. You know, suppose this is more informative information. I have the gene ID or transcript ID and the section of the gene. Even the section of the genes, they will, you know, tell you what is the overlap, the distance. What is the percentage? Sometimes a transcript is bigger than the peak, sometimes the other way, or sometimes they all half overlap. So this is the table. You can download this table as text file. Once you have this information, one way to look at each sample individually is to run a TSS plot. So under exploratory analysis, you run TSS plot. Basically, we line up all the genes at the transcription start site. So this is the TSS. We go upstream, 1,000 base pair, downstream, 1,000 base pair. This is determined when you perform annotation. If you say 1,000 is too long, I only want to look at a 500 base pair, you can do so, okay? And then, so this each line is a sample, and we can see pretty much everybody is a little bit downstream of the TSS for all the samples. And the bottom is the heat map. The heat map by default is each row is one sample. It's not very pretty. You can change the heat map per row. If I say I got 6 per row or even 8 per row, I think it's better to look at this way. So for the heat map, it's the same information as the profile, but instead each row is a gene. We kind of sorted the gene based on the read. In this location, you know, TSS up and down 1,000 base pair, and we kind of sorted them, and you will, it's a different visualization to see the enriched region. And sometimes the color, probably the color scale is not very clear. You can change the scale. So suppose I use maybe 100, and to see, no, this is maybe too much, maybe 500. So you can adjust this color to see which one, you know, look nicer. It's very much the same story, but different visualization. Okay, so for each sample, this is, you look at each sample individually. If you have a ChIP-seq, so for the ChIP sample, and when you display the input sample, you don't see this pattern. You don't see the enrich. They don't enrich anywhere, right? Okay, so this is one way. And the other way for the comparison, suppose you only have 2 samples or 3 samples, you don't have a replicate for different treatment. You just want to see, okay, which region is common for all the samples, which region is unique in one sample. So only if you don't have a replicate, I would recommend you to perform the compare regions. There's nothing to configure. You click on that. This result will compare all your samples, all the regions. Basically, is you know, all the breakpoint. We just join all the breakpoint of all the regions and give you the report. Say, okay, this region. You can see when you chop it up, the read length, sometimes maybe this is just a noise, right? This one base pair is happening in this one sample. Probably is because this sample we detect a little bit longer than the other. And then next to that, we got 3, it's only 3 base pair. There are 4 samples out of 16 detected, right? So when you do this kind of comparison, you kind of chop up your fragments is very fine. If that is the case, what you can do also, if you say, oh, either you can filter out all those, you know, a few base pair, only focus on the bigger base pair. Okay, here, all the 16 samples they detected, probably they are the same region. And some sample, if you look at this, some is a little longer, some is a little shorter, right? Depends on what your goal is. If you only want to look at the bigger picture, you can just filter out all the other, only look at this one. Okay, and after you filter that, you will look at, okay, if I look at a 200 base pair and this region only happened in one sample, that probably is unique in the one sample. So this compared region, you can perform filter, and then you can filter out some read length is really low. Okay, to get to either, and then you look at the table, look at the unique or the common region, etc. Another thing is, if you combine that, suppose you only have 2 or 3 samples, only very few number of samples, you will do that. And if you say, I really want to look at the bigger picture instead of look at the common region, like here, bigger region, I want to look at the bigger one, or which one is the longest one, we do have this function allowed you to merge regions. So this merge regions allowed you to combine the region if they are next to each other. So let's take a look at this guy. And here you can see the stop of here is the start of this guy, right? This one, the stop is the start of this one. So suppose this is really one region, or eventually, if I got to over here, probably all this is one big region. Suppose you, I cannot select it. From here to here, I think it's one region. Instead of, I want to look at 361 base pair, I want to look at like a 380 base pair. So you can merge that. You can, if you say, I only want to merge the regions, there's no gap. The previous stop is the next one's start. You can put the 0 here. But if you say, I will allow there's a little gap here, sometimes, especially when you look at the peaks, some peaks. So for example, I just look at the, suppose those 2 peaks. If I want to merge them together, I will allow a little gap here. I think your gap, the longest one, should be less than your read length, right? Your read is your unit of measuring. You shouldn't be longer than that. If the gap is longer than the reads, or twice or 3 times, that probably is a separate peaks. But you can define. So I will merge if the gap is less than 5 base pair or 100 base pair. So this function depends on what you, sometimes you will answer a question, on this. All right, so this is when you only have a few number of samples, what you can do on the peaks. In our case, we have a tumor versus normal. I do have 8 replicates for each phenotype, and I want to do the comparison. Give me some statistics. If that's the case, you need to quantify the regions. So basically, if you want to cross a sample to do the comparison, you have to make all the regions, they line up for every sample, just like genes, right? So we will perform quantify regions. Sorry. And when you quantify the regions, so first of all, we're going to join all the breakpoints. So like here, one big, let me just zoom in a little more. So if you have one sample of the region is from base pair 1 to 10, the other one is from 8 to 15, we're going to make the regions 1 to 8, 8 to 10, 10 to 15. So we just join all the breakpoint, and then we count. So that way, all the samples will have the same start and stop for all the regions. And then we can calculate how many reads are in this region. And then if you have a peak here, you're going to have more reads for that sample, right? So that way we can compare across samples. Now let's take a look at the quantified region dialog. Here you have to define what is the minimum region. Once we join the breakpoint, like what we saw, sometimes you only have 1 or 2 base pair. I don't want to look at that. You will say, okay, my region should be at least such and such. And I would think your region should be longer than the read length, right? Like your fragment. If you know your fragment length, suppose it's 150 or 100. If you want to be more permissive, I would say, okay, I will use 110 because my read length is 100 already. And also, you want to see, okay, how you want to count this read belong to this region. Like when we do RNA, we will see your read has to be fully within the exonic region because you extract the RNA, but here peak is kind of noisy. So I will allow some overlapping, hanging there. If you want to be stringent, I would say, okay, if the read is 90% overlap with the region, I would call this region belong to the region. Or you can be a little bit more permissive. Say if half of the reads, overlap with the region. Oh, I'm sorry. I zoom out. I want to zoom in. Okay. So now you can see once you zoom out, this is a bigger picture. Once you zoom in, you can click and drag, that will zoom in. Close this guy. Okay, so, and then after we assign the reads, again, just like RNA-seq, you can filter out to low. If a region, the total read number is less than 10, I can filter that out. Okay, so you finish that. And from here, you probably want to re-annotate the region because the peak over here and quantify is a different peak. Here we over segment that, so you want to do the annotation over here. It's the same dialog. We just get the gene information related to the peak, and then you can also perform the filter. Yeah, so this is the region annotation. You can also perform filter low read count here. I think a quantified region will give you a table. This table is very much like the quantification for RNA-seq. Again, the minimum is going to be 0. And then you can see some reads you have a 200. Some regions you have a 200 reads. And then you can use the median to try to perform a little filter here. If you download this table, it's just like your RNA-seq, but instead each row, we can go ahead and download this guy. Put the features on column, on rows. So we can open that in Excel. Now, in each row is a region. We have a start and stop, and actually the ID is the region, chromosome start and stop, and this is the read count for every sample. So now it's a matrix. Every sample we can do the comparison, right? So from here, the downstream analysis, if you want to detect differentially enriched region between tumor versus normal, it's going to be very much the same as your RNA-seq. So you're going to perform normalization, and I use the same TMM log because later on I want to do the correlation, and then use Illumina to generate a significant region. So everything is the same. Now the result will be, I have a region, that's the region ID. And this, once you do annotation, you will have annotation information. If you don't do annotation, you won't have that. And the annotation, we have all those information. Every time in the optional column, you check on things here, you will make the table bigger. They will just add more information, okay? I want to add less because the transcript ID, I don't know what that means. I will just keep the gene ID, and I know. Oh, when I filtered based on the only the TSS region, and then tumor versus normal, code change, p-value, etc. Okay, so this is the significant regions I cared about, and also you can do dot plot or heat map. Is the same thing. Okay, so I think it looks pretty good. Do show the tumor versus normal in those regions, they are different, right? So, and also because we add annotation, we add a gene name here. As long as you have a gene name, you can perform enrichment analysis. You cannot perform a GSEA because GSEA needs to know the gene expression value. But here we don't have a gene expression value. We'll only have the gene gene name here. We count the gene name. We can look at the pathway enrichment. Okay. Another special thing you want to do for ATAC-seq or ChIP-seq once you have a significant region is, I want to look at those regions, what is the motif there? What is consensus sequence there, right? So you can click on that and do a motif detection. Either you can detect a motif, a novel motif, and you have to specify the length. This is a long process. So typically, suppose you only look at one type of antibody, you probably only detect one motif. If you have multiple, it's going to be really messy. I would recommend you do one at a time. And the result will give you the sequence logo here. So this is what I detected in those regions. This is the consensus sequence. And the detail will tell you we found in which location. So those are the location we found this sequence. The other one is, since the pipeline is really long, let me make it shorter. So you can collapse the pipeline. If I right click on this, we can collapse this pipeline. I just put it over here, make it shorter so I don't have to scroll to the right. This is the collapse, so you can double click to open it. And this, that means you just remove this collapse. It's not a deleted task, okay? So I'll just recollapse that. Just a little tip here. And the other one is I want to detect known motifs. And we do have a JASPAR database, and this is a human. I will choose a vertebrate database and click finish. That will give you this known motif result. I think everybody will match this guy because this is too general. And if you click on any of this motif, that will take you to the JASPAR database. Okay. And again, so where we found which motif, and that is the location. So what is the motif ID and the sequence of the motif? And you can use a search to look at the location. So this is for ATAC-seq and pretty much what you're gonna do. Basically, you do peak detection and annotate peaks. And then once you find significant regions, you can detect motif. Okay, so the last part is how we integrate those two assays. One thing is, suppose I got the significant regions, and within this region for ATAC, I do have a gene name here. And if you say, okay, so this region, suppose they are in the TSS of the genes, I want to see how these genes, their expression value look like, right? So one thing you can do is you can use a Venn diagram. Venn diagram just compare the name, right? So I will look at my gene list. This is my significant gene list. Make sure you choose the ID you want. We have an ensemble ID, or I can use the gene name. And also I want to compare, this is my significant list of ATAC. And their ID is a region ID, but I don't want to compare region. I want to look at the gene name. And once you select that, you can display that. So this is the common genes. I'm not sure of the biology there. And this is I forgot to change the name, but you can make a name. This is the ATAC. This is the gene. Okay. And this is the GX. I know the color. That's why I know how to change that. So this is the unique genes. So every time when you click, you have a gene list, and you can download it. or you can save that to your list. This is download. You can save that to a list management to give a name, and later on you can use that to filter your other project. Or you can use this genes to say, hey, I want to look at the ATAC unique genes. I want to see how this gene expression look like, right? So I get this gene. You can click on this filter, say, I want to filter based on the normalized count, or I can filter based on my significant genes. It doesn't matter where you want to do. And make sure you're going to filter based on the gene name. The key they should match. So if I do it this way, those, this gene list, I filter based on the gene expression, this normalized count, you can perform PCA to look at the data and then perform, if you perform on the differential analysis, you can get the gene list to see how those genes, p-value, et cetera, look like. So this is one way. The other way is, think about this situation. I have 16 samples. Each row is a sample. If I want to look at, one region, how they correlate with which gene they highly correlated, that probably means, this binding site will regulate that gene, right? So we do have this function called to correlate between two assays. However, when you perform this kind of correlation, you should choose a count matrix. You have to have a sample, sample by feature matrix, normalized count. But this is too many. I got, 100,000, 17,000 features. This one is 37,000 features. If you want to correlate everyone from here versus everyone from, that's huge. You probably don't care some genes you don't care. So I would highly recommend you to perform filter on the data. You either, get some significant gene list to filter the data. Here, I just use some example to filter this matrix down to 200, almost 300 genes, the significant genes. And this one, I also filtered down to, 400 features. So this is a subset, less number of the features, but the sample size is still the same. So you select one of the data nodes, you will perform statistics correlation. And this correlation is called correlation across assays. Basically, you perform a correlation between two different data nodes. And then it will ask you, okay, what is the other data node? I will select this one. So we will look at every features in one data node compared to every feature in the other data node, okay? And you have to pair them up for every sample, how you pair them up. Sometimes when you do a single cell, every cell, they already paired up. You can use the sample name because the barcode, they will pair. But for this one, I do have the assay pair. So the same sample, you have to label them the same. Okay, with the 32 samples in total, you're going to have 16 pairs. So this is the key for you to perform this kind of correlation. You can perform, you say, I want to look at every feature in one node versus every feature in the other node. You can do it this way. Or if you say reduce the correlation, I only look at the genes with correlated with the regions on the same chromosome. So that way we'll highly reduce the pair up. And then when you do the correlation, you can do Pearson correlation or rank correlation. And you can choose to report, only report if the p-value is less than this, and the correlation coefficient is either up and down, the absolute value is greater than 80 or 85, 0.8 or 0.9 or whatever your cutoff is. So click finish. This result will have this correlation pair. And that will tell you, okay, the feature one is the first data node you select, and the feature two is the other one. You can add optional. There's not a whole lot. Feature one is my region. Oh, no, feature one is my gene. You can see the gene ID and the gene name. Feature two is the region, and the region, the next to which gene, right? So we only report the TSS of the gene. And then this is the correlation coefficient. You can sort it this way up or down, negative correlation and positive correlation. And whenever you see a correlation, and this is the correlation plot. So you look at the intensity, x-axis is my gene expression value. So we select the normalized count, right? So the normalized expression value of the gene. Each dot is a sample. I have 32 dots here. And then the y-axis is the region, and that's the normalized read count in the region. So this is the correlation coefficient. Currently, we don't have the chromosome view there yet to directly invoke that. But in the future, we're going to add that. But if you funnel down to certain genes or regions, you can open your chromosome view. And then, you can search for this location and add a track of your gene information and add a track of your transcript. Because we run out of time, let me just show you a trick here. Once you close a couple of tabs here, I can save, if you want this region, you want to look at this again, we have a bookmark. You can always bookmark this, or there's one called, you can send it to the notebook page to save it. So I saved one in the notebook page, the notebook, not data viewer. You click on this, and this is what I look for the region. And in this notebook, the picture is dead, but click on this button, will say back to the source. That will make it live. And this is a region I found that, they have a high correlation of the gene. So this track, the blue track, is my star alignment of the gene expression data. I use two color represent tumor versus normal. Blue is normal and the tumor is red. So we can clearly see normal sample. I have more reads. And this is you can see the ensemble annotation, and this is the transcript, but I calculated the gene level. So this is the gene location. And on the top is my ATAC-seq data. I can clearly see the normal is high and the tumor is low. And this is kind of in the promoter region, right? So over here, so suppose that this is transcription site. Some is from here, some is from here, but up and down 1,000 base pair, probably this is the your promoter region. So this is kind of related. So this is another way for you to present your result for this particular gene. All right. To sum up, this data, if you have this type of the data, usually it's a very big experiment design. And to perform a correlation, you have to have one, you have the same sample, you extract the RNA and the DNA, you perform on two different assays. Otherwise, you cannot do the correlation analysis. So you can compare the Venn diagram, if they are completely different sample, you can also, do those two branches. And once you have a gene list, use the Venn diagram to compare the genes. All right. Now I'll be happy to take questions.
Thank you, Shaolin. There isn't any questions in the chat. But there is one that asks, can Partek flow be used to analyze single-cell RNA and single-cell ATAC data?
Yeah.
And I responded yes to that, if that's correct.
Yes, thank you. Yeah, I think in 2 weeks we're gonna have a webinar talking about a single cell RNA-seq and ATAC-seq in Partek.
Okay. Right. Okay, so I've dropped the link to the BTEP calendar
Thank you.
into the chat, and where they can find out more information about the upcoming class.